Hello, this is a virtual poster presentation for the Undergraduate Research Symposium. We are Research Team 3 from the Purdue course ABE 227. Individually, we are Amanda Ventura, Rebecca Slaughter, Ben Burns, and Antonia Divick. Our research topic is genetic similarities in phage cocktails targeting Staphylococcus aureus and avian E. coli. In the past 30 years, phage therapy has emerged as a promising new treatment for bacterial infections that is an alternative to ordinary antibiotic treatment. Phage therapy uses bacterial viruses, known as bacteriophages, to treat infections. Bacteriophages are a potential solution to antibiotic resistance, as they can adapt to bacteria's resistance over time. To best utilize phages and to overcome bacterial resistance to individual phages, phage therapy often uses what is known as a phage cocktail. A phage cocktail is a mix of multiple phages that infect either the same host or different hosts, depending on their intended use. The most successful cocktails are able to lyse bacteria quickly and adapt to any mutations of their bacterial target. The first step of our process was to collect research papers that tested different phage cocktails for the same host. These cocktails were made up of multiple unique phages, and we selected papers with phages that were annotated and accessible on NCBI GenBank. Cocktails we chose were successfully able to significantly reduce the population of host bacteria. The genomes of the phages in these cocktails were compared in DNA Master using the genome comparison tool to create a gene map. This can be seen in figures 1 and 2. First, the genomes of the phages in each individual cocktail were compared. Then, the genomes of all the phages found were compared to each other. DNA Master also generated a draft evolutionary tree that shows, if these phages were all of a known family, their evolutionary closeness. From our genome analysis, we had 3 main points. Within cocktails, there was relatively low genomic similarity. Many of our comparisons show that phages within the same cocktail had very few genes that were homologous to each other. However, when genes from different cocktails were compared, more similarities between the genomes emerged. This leads us to main point 2. Some phages from our cocktails showed high genomic similarity to phages from completely different cocktails. This suggests that, although there are few similarities between phages in the same cocktail, phage cocktails attacking the same bacteria have phages that were genomically similar. Main point 3. The DNA Master draft phylogenetic tree further supports the similarity between cocktails by grouping phages from different cocktails together and separating phages from the same cocktail into different groups. These results may be useful in informing future phage cocktail design. Currently, many researchers, by analyzing effective cocktails genomically, we were able to identify certain trends between cocktails. These trends may help inform future phage cocktails that are attempting to infect the same bacteria. Since we found similar genes across phages from different cocktails that infected the same bacteria, a future phage cocktail researcher might choose a phage that is also genomically similar to these phages, as it seemed to be a common feature of the successful cocktails. Potentially, this research could begin a foundation of phage cocktail design that relies on a bioinformatic approach rather than simply experimental methods. In terms of further study, even though our DNA Master result highlighted homologous genes, further research has to be conducted to determine the function of these genes and perhaps how this function relates to the phage's infection mechanisms. To further validate the comparisons that were found, more well-documented bacteriophage genomes for the host Staphylococcus aureus and avian E. coli need to be collected. To assure the results are consistent across hosts, more bacterial hosts should be analyzed. We identified Klebsiella pneumoniae as a potential future host to be compared due to the large number of annotated phage genomes from phage cocktails that were tested on it. It would also be useful to further define the success of a phage cocktail. So when analyzing phage cocktail characteristics, we can determine which characteristics are associated with which successful cocktails and which characteristics are associated with less successful cocktails. Ultimately, more bacteriophage genomes will be required to provide substantial evidence to support any claim from this research. Thank you.